Hi everyone, today I'll be explaining shortly about the mitosis and meiosis. So first I need you to know about the cell cycle. The cell cycle consists of two main stages, the interphase, which is from this to this, and the mitotic phase, which we will discuss soon. Interphase is the longest part of the cell cycle and is divided into G1, S, and G2 phase. During G1, the cell grows and carries out its normal function. And in the S phase, its DNA replicates, so each chromosome is duplicated. And by the time the cell reaches the G2 phase, it has fully replicated its DNA and continues to grow while producing the proteins and organelles needed for cell division. The G2 phase acts like a final checkpoint to ensure that DNA replication has occurred correctly and that the cell is ready to enter the mitotic phase, where mitosis and cytokinesis take place. So I'll start to explain from the G2 of interphase. So as we can see, a nuclear envelope encloses the nucleus, and it also contains nucleolus. And we have these two centrosomes, which is duplicated from one centrosome. And centrosomes are region in animal cells that organize the microtubules of the spindle. Each centrosome contains two centrioles, which is perpendicular to each other. Yeah, it's just additional information. Chromosomes duplicated during S phase cannot be seen individually because they have not condensed. They are still in this uncondensed form. So next, to the first stage of mitotic phase, which is the prophase. It's actually easy to memorize it by just memorizing PMAT. So prophase, metaphase, anaphase, and telophase. I memorize it by using that PMAT. Anyway, so the prophase state, the chromatin fibers become more tightly coiled, condensing into discrete chromosomes observable with a light microscope. So, and the nucleoli also disappear in this state, and each duplicated chromosomes appear as two identical chromatids, and it's joined at their centromeres. This is the centromeres. It's like the part where they join. It's almost in the middle, but sometimes not always in the middle. Yeah, not always in the middle. And the mitotic spindle begins to form. This is the mitotic spindle. It composed of the centrosomes and the microtubules that extend from them. And this is called aster because it looks like a star, and aster means star. Okay, so next, between the prophase and the metaphase, there's a transition phase called the prometaphase. Some book explain this, but some don't. I take this from Campbell, and yeah, they explain prometaphase, but I don't think in Cambridge they need this. But I'll just be explaining this stage. So the nuclear envelope fragments, yeah, almost. So this fragment, it doesn't form like this sphere anymore, and the microtubules extend from each centrosomes can now invade the nuclear area and connect with the centromeres. Oh, I think it's so it's like this. The aster, this is the star, and so the kinetochore just started to attach to the centromeres and make this chromosome align in one plate. It's called the metaphase plate, which is actually imaginary. And some kinetochores that doesn't attach to chromosomes overlap. It's called overlapping non-kinetochore microtubules. This is just so that you have a better understanding. It's not really important. Okay, so on to the next phase, the metaphase. So the centrosomes are now at opposite poles of cells. The chromosomes have all arrived at the metaphase plate, a plane that is equidistant between the spindles of two poles, and the chromosome centromeres lies at the metaphase plate. For each chromosomes, the kinetochore of the sister chromatids are attached to kinetochore microtubules coming from opposite poles. Next, in the anaphase, it is the shortest stage of mitosis. It's pretty quick because you just need to drag this to the opposite way. Anaphase begins when the cohesin proteins are cleaved. This allows the two sister chromatids of each pair to part suddenly, and the cell elongates it's kind of elongate compared to this one, right? And then telophase, the nuclear envelope start to forms and cleavage furrow forms. It is also, there's like contractile ring made of microtubules. Oh no, sorry, intermediate filament. So the ring contract and separate these two into two daughter cells. Now that you understand mitosis, I think it's pretty easy to understand meiosis because it's almost the same thing. So this is mitosis. There's like a parent cell. This is 2N or diploid, and then in metaphase, it aligns and separates into two daughter cells, which all contains the same genetic material. So it's basically identical. Meanwhile, meiosis, the first meiosis, it separates two homologous chromosomes. So we are diploid. We contain two sets of chromosomes. So the homologous chromosomes just align in this metaphase, and it separates into two daughter cells, which is a haploid because it lost its pair. And meiosis 2 is like almost the same as mitosis. It separates the sister chromatids and form this daughter cells. And if you can see, it's not the red and blue is not the same, it's not identical. So non-identical daughter cells, because they experience crossover at the chiasmata. Ini in this stage there is protein called cohesin that connects them, and it allows crossover to occur, and so they form this chiasmata, and that's why the cell can form crossover. Yeah. Selesai. Thank you.